Good morning students, my name is Jagyasani Samanta and here today we are going to discuss about a very simple and easy topic, that is your cell cycle and cell division. So most of us know that with the growing of our age or day-to-day life, the body parts also grow, right? So if you can consider about your height, weight, 2 year or 3 year ago, the height was not same or the weight was not same. It changes every day-to-day life, right? So if anyone questions you why the height or weight of our body changes, so the simple answer will be cell cycle and cell division. So the growth of the height or the weight gaining process is basically the process of cell increase, cell number increase in our body. Okay, so that is our cell cycle. So how the cell gonna increase the number in our body, and that process is known as cell cycle. So today we are going to discuss about the entire and in detail about the cell cycle process, how it occurs, what are the phases and what is its importance. Okay, so let's start. So this is our first topic, is cell cycle and cell division. So obviously there is a question arise, so what is the difference between cell cycle and cell division, because we are studying two of the terms at a time. So cell division is the process or it is the part of cell cycle. Okay. So we are going to discuss first cell cycle. So cell cycle is a very common process. All of you must hear the term mitosis meiosis, right? All of us heard in our school time. Okay. So the mitosis meiosis is the cell division process. Okay. And this cell division is the part of your cell cycle. Okay. So now we are going to discuss the entire cell cycle process. So basically the entire cell cycle process is a two-step, two-phase. Okay, one is I phase, second one is M phase. So I phase stands for, I stands for interphase, M stands for mitotic phase. Okay, so I stands for interphase, M stands for mitotic phase. Okay, so what is interphase, what is mitotic phase? So interphase is also known as preparatory phase. It is also known as preparatory phase. Then question arises, why it is called as preparatory phase? Because in the mitotic phase we found out two steps. It is in a two types of process. One is your mitosis and second one is your meiosis. Okay. A cell, one mother cell gave rise to two daughter cells. All of you know this. Okay. So this one mother cell give rise to daughter cell. It basically divide itself into two equal halves, right? So this is a simple concept. A one mother cell divide itself into two equal halves, which are known as two daughter cells, and this is known as your M phase or mitotic phase. It occurs either by mitosis or meiosis. We will discuss this later. But in order to perform the mitosis or meiosis, that cell has to go through before phase, that is your preparatory phase or interphase. Then why preparatory phase is important or why it is necessary? The thing is, when a mother cell divide itself, okay, in mitosis and meiosis, all of you know this mitosis is an equational division, right? So the number of chromosomes in a mother cell is equal to the number of daughter cell. Number of chromosomes in daughter cell as well, right? Then how it occurs? By this process, interphase. So basically preparatory phase is the time period of cell cycle where it prepares a single cell to get ready, or it get ready is the cell for the mitosis and meiosis division, right? So that's why it is also known as preparatory phase. The entire energy, the RNA, DNA, organelles, whatever the cell require in mitotic phase, all things will be ready or prepared during this I phase. That's why this is called as preparatory phase. Then the question arises, how much time it takes? If we consider a cell cycle, in a human cell it is average 24-hour cycle. One cell cycle complete in average 24 hours. Okay. So in 24 hours, 90 to 95% time. Okay. So 90 to 95% of time was occupied by interphase. That is equivalent to 23 hours. So if a cell goes through the cell cycle process, the cell will remain in interphase for 23 hours. Only 1 hour it will require for to complete the mitotic phase. Okay. Then obviously why it takes such a long time? So the answer is the interphase is not a single step, it is in a 3 phases process. First one is G1 phase, second is S phase, and third is G2 phase. Okay. In interphase, there are 3 subphases. First is G1 phase, second is S phase, third is G2 phase. So now we're gonna discuss in detail about each and every phase. Okay. So let's discuss about G1 phase now. So in J1 phase, the first and foremost thing occur is production of ATP molecule, because cell has to go through the mitosis and meiosis process. Obviously, it requires some amount of energy, and the energy which is required for the cell during mitosis and meiosis process, that was produced in preparatory phase. If someone asks in which phase or in which time the cell produced the entire energy, the answer will be G1 phase. So first thing occurs is the production of ATP molecules. Okay? The next is the synthesis of RNA, because in cell RNA is really important. That might be rRNA, mRNA, tRNA. There are so many things inside this. So the synthesis of RNA, proteins are also occur in the G1 phase. So the synthesis of protein, synthesis of RNA, and the synthesis of ATP all are occur in this G1 phase. So there's a little thing. Just think about it. If RNA, protein, ATP all are synthesizing in G1 phase, the cell size will be increased. Definitely yes. The cell size will also increase during the G1 phase. When we call about or we look about the phases or the work occurs during the G1 phase, like ATP production and synthesis of RNA and protein, cell size increase. So it's ultimately leading to the point that this in G1 phase, cell is highly metabolically active, right? This is the NEET question in 2017. This question has seen. In G1 phase, the cell is highly active. Yeah, the metabolic rate of the cell is high during G1 phase. Okay, so this is all about your G1 phase. This is the initial or first phase of your interphase. In this phase, the ATP productions will be occur. The ATP production, why the ATP are required for the next phases like S phase, G2 phase, mitosis, meiosis, for the entire cell cycle, the amount of energy which are required are produced during G1 phase. Then second thing, synthesis of protein and RNA are occur during G1 phase. Then third, in this phase, due to the synthesis of RNA molecules, proteins, the cell size obviously increase. Then last, which is a very important term, it is actually was a question in 2018, 2017-18 question. This is in which phase, in NEET exam, in which phase the cell is highly metabolically active. Okay, so the answer will be G1 phase. Okay, so this is about your G1 phase. I hope it's clear. So now we're going to discuss about S phase. Okay, so next is your S phase. So S phase, you can remember it as a synthesis phase. Okay, what it synthesis in S phase, the most important terms, the most important part of the cell, that is your DNA. DNA is synthesis during S phase. In DNA synthesis, in order to package the DNA, along with DNA, histone proteins are also synthesized during this phase. Okay. So in S phase, the most important work is done, that is the synthesis of DNA. In order to pack the DNA, along with it, the histone protein also synthesis. Along with these two, during S phase, a centriole also produced. Centriole duplication also occurs, which means a single centriole produce two centrioles, which gonna be help in mitosis and meiosis division. So basically the cell performing all the activity in order getting ready for mitosis and meiosis. Okay, so this is a simple basic topic. So mitosis, meiosis is in a cell division process. Okay, one cell will give rise to two daughter cells. So whatever things required for the division, all are getting ready in this phase. Okay, so first we have discussed about G1 phase, where the ATP molecule is produced, synthesis of DNA, RNA and proteins are done. Cell size will increase, and due to all this we can say that the cell is highly metabolically high. Okay, the metabolic rate is really high in this step. The next S phase is a synthesis phase, which is very important. Why? Because during this phase, the DNA, which is the master of your cell, and in order to pack the DNA, histone protein and central duplication occurs. Before going to the G2 phase, there is a small numerical questions comes in NEET exam. So we are going to discuss about that. Okay, so the numerical questions will be regarding your C value and N value. Okay, then what is C value, what is N value? So basically N value stands for the number of chromosomes. Okay, so N value stands for what? Number of chromosomes inside the cell. In C value, C stands for the total content of DNA inside the cell. Okay, so what is the difference between C value and N value? The first thing is, this is the number of chromosomes. Okay, and in C value, the content of DNA. Okay, so how we will describe or how we will differentiate between the chromosome and DNA? Now let's do that. It is simple. How many chromosomes are there? Obviously, answer is 3. So the N value is 3. Okay. And here, if I comes to what is the C value? Again, it is 3. Okay. But when the cell completes its G1 phase, enter to S phase, what will occur? The synthesis of DNA will occur. Okay, so it's not the synthesis of chromosome. What is doing in S phase? We already discussed, that is the synthesis of DNA, which means the amount of DNA is increasing, not the number of chromosomes. Okay, so let's do that. So the content of DNA is increasing, it's synthesizing, it's basically making it double. So if we discuss in a cell, the N value is 3 and C value is also 3 in J1 phase. Okay, after completion of S phase, what will be the N value and C value? Okay, if it is the question, basically a simple, very simple question. So in a cell, if the N value is 3 and C value is 3 during G1 phase, okay, what will be the N value and C value after completion of S phase of that cell? Okay, so let's do. All we know, in S phase what will occur? Synthesis of DNA will occur. So number of chromosomes remain same, nothing going to be changed. So the N value remains same, that is a 3. But the C, which is the content of DNA, DNA is what? Synthesizing in S phase, is making it double. So just to what is the C value during G1 phase? 3. So it's making it double. So 3 into 2. Basically the C value will be 6. This is a simple thing. Basically here you have to understood is, after the completion of S phase, the C value will increase. The N value will remain constant in each and every phase. N value remain the same. That's nothing going to be changed in none of the phase. Okay. So the value which is changing, that is your C value, and when it will change? After the completion of S phase. When the S phase is completed, the number of or the content of DNA becomes double. Okay, this is a one-time process. After the in the interphase, in S phase, the content of DNA will be double, and after that it remain constant. Nothing gonna be changed. Okay, so only thing you have to remember is after the completion of S phase, the C value will be double. And if you ask a question, ma'am, what if SL has N value 3 and C value also 3 during S phase or after completion of S phase? Then what will be the N value and C value during the G2 phase? Very simple, is nothing has to be changed. The N value remain constant, we all of you knows that. So N value remain also 3. Okay, and the question it is mentioned after the completion of S phase the C value is 3, which means the duplication of DNA or the synthesis of DNA is done. So after the synthesis of DNA the value is 3. So nothing gonna be changed. In G2 phase the C value will be again normally 3. Okay, so nothing gonna be changed. I hope N value and C value are clear. So let's make it more clear. Let's do some more questions regarding this. Okay, so basically let's take a question. SL has C value 20. Basically C value is the measuring unit is picogram, PG, picogram. Okay, so the C value is 20. Then find out the C value of endosperm and egg. Okay, so what is the question is SL has C value 20 picogram. Okay, find out the C value of endosperm and your egg. Okay, so obviously the question comes to your mind, how we will do this question, because we have studied about this in G1, S1, G2 phase, but here the topic, here the content is totally different. So the answer is simple. You have to just simply know about the ploidy stage. So a cell means it is in a diploid state, which means 2n equals to 20 picogram. So 2n is the total content of DNA. We all know that the number of chromosomes is containing the number of DNAs. Okay, so the total content of DNA in cell is 2n, which is also known as diploid. So which means in a cell a chromosome contains the total number of DNAs. Okay, and the total number of DNA, how much is given, is 20 picogram. Done. So we know that 2n equals to 20 picogram. Okay. Endosperm is in a triploid stage, which means 3n, and egg is in a haploid stage. Okay. So now let's come to the C value. Then obviously many of students get confused here with the N and C value. Let me clear you something. This is the 2n. 2n means total number of your chromosome. The total number of chromosome contains the total number of DNA. Okay, the total content of DNA. So basically this 2n or this 2n stands for C equals to 20 picogram. Okay, don't get confused this n with this n value. No, that's not this is the number of chromosomes. Okay, here it has given clearly in picogram. Picogram unit is the C value unit. Okay, so here we get a C value. So C value stands 2n equals to here 20 picogram. Okay, so basically n equals to 20 divided by 2, which is 10 picogram. So the C value of egg is 10 picogram. And endosperm is a triploid state. So obviously n value we know 10. So 3 into 10, 30 picogram. So 30 picogram is the endosperm structure, endosperm C value. Okay. I hope this thing is clear. And if you want to make it more clear, then just in phases if you want to talk about. So the n value constant in G1, S, G2 mitotic meiotic phase, nothing going to be changed. Mitosis, meiosis, after the end of the meiosis 1, it's become reduced. Okay. But in G1, S and G2 phase, the n value remain constant. The C value will change after the completion of S phase. There's 2 points you have to remember to solve this kind of questions. If you are clear with this, you're going to clear all the numericals. There's nothing to worry about it. Okay. So now come to our G2 phase.